Meiosis is the division and creation of egg and sperm cells or gametes by separating homologous chromosomes. It is a two-part process containing interphase 1 and 2, prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, and telophase/cytokinesis 1 and 2. To begin, when the cell is preparing to divide, it goes into interphase 1, a multi-step process where the DNA is replicated. First is G1 or gap 1 phase, where the majority of growth takes place and new proteins and organelles are synthesized. It is then possible, although it doesn't always happen, for a cell to enter G0, which is when the cell cycle halts due to unstable environments or a cell's inability to divide. Next is S phase, where the DNA is synthesized and copied, resulting in double the amount of DNA. Lastly is the G2 gap phase, where many more needed organelles and proteins are made where the centrioles are copied. In addition, the chromatin within the nucleus of the cell begins to wrap tightly around proteins called histones, starting the creation of the chromosomes. At this point, meiosis 1 occurs. The first step is prophase 1. During prophase 1, the chromosomes are first visible and the spindle fibers are created. Homologous pairs or pairs of chromosomes, one from each parent, are formed from the new chromosomes, making tetrads. Then crossing over occurs, which is when homologous chromosomes exchange portions of chromatid, producing a new combination of genes. This is where genetic variability comes into play, because now there's 4 different chromosomes, all with random different alleles for each gene, a random combination of the mother and father's genes. Next is metaphase 1, where the tetrads of the homologous pairs line up along the middle of the cell, remaining in pairs, and the spindle fibers attached to the centromeres. After this is anaphase 1, where the homologous pairs are pulled to opposite sides of the cell by spindle fibers. The last step in meiosis 1 is telophase and cytokinesis 1, where the homologous chromosomes split into 2 daughter cells. This occurs when the cytoplasm divides and the cell membrane pinches in at the cleavage furrow to create 2 haploid daughter cells. In addition, the nucleolus and nuclear membrane reform. The second part of meiosis is another multi-step process called meiosis 2. In this part, some cells skip interphase 2, although others only perform G1 and G2, since there is no S phase in meiosis 2. In prophase 2, the nucleolus and membrane break down, and the chromosomes are already coiled from the previous round of division. Spindle fibers form and move to opposite sides of the cell. Next is metaphase 2, where the individual chromosomes line up in the middle of the cell. The spindle fibers attach to each chromosome centromere. In anaphase 2, sister chromatids split and are pulled to opposite sides of the cell by spindle fibers. During the last step of meiosis 2, the previous 2 haploid cells divide into 4 new haploid cells that are all genetically different. The cytoplasm splits and the cell membrane pinches. The spindles break down, the chromosomes uncoil, and the nuclear membrane then reforms. At the end of the process, in males, 4 sperm cells are created, all capable of reproduction. In females, however, the cell splits unevenly, and only one of the 4 is large enough and capable enough to support life. This is called the ovum, whereas the other 3 smaller eggs are known as polar bodies.